Welcome to the last webinar of the year 2020. If you want to present a talk next year, you can send us an email at the email address that you can see on your screen. And you can also send us an email if you're interested in getting involved in yICSA, because we will have some positions open soon. So today we are welcoming Dr. Johannes Grillari from the Ludwig Boltzmann Institute of Experimental and Clinical Traumatology in Vienna, Austria. And Dr. Grillari is the director of this institute, and is interested in RNA biology and senescence and aging diseases. So today, Dr. Grillari will talk about extracellular vesicles and microRNAs in cellular senescence and age-associated diseases. So you can share your screen now, or I can stop sharing. And unmute yourself.
I think that should work now. Yeah, so dear Alice, many thanks for the kind invitation to this webinar. It's a pleasure to be here. It's also fantastic to hear that there is young ICSA, so I think that's a fantastic move of your side. Really appreciate it, and the more it's a pleasure and honor to present here. Thanks for the kind introduction as well. So besides being at the LBI for experimental and clinical traumatology, I'm also still at the University of Natural Resources and Life Sciences in Vienna. And as to make clear any potential conflicts, I'm a co-founder of two companies at the moment, Tamerna and Eversight. And so with that, I think I go to the next slide, and it's what we all want to know. What are the drivers of biological aging? And my lab has been focusing, and that's why I'm here, on cell senescence for a pretty long time. And I think we have here two very nice examples of the different speed of aging. Tina Turner photo was taken at almost the same time as the photo here of Keith Richards at the age of around 65. And we think that this guy probably is exposed to a high load of senescent cells internally, and probably this is due to some stress-induced premature senescence. I think the toxins running in the veins of Keith Richards, they might have contributed. On the other hand, we have probably here some extrinsic rejuvenation, probably not senolytic, more the knife type, but still. So I think I don't need to expand on this. We know that chronically accumulating senescent cells are detected throughout the human body in a lot of different organs, and in many instances correlated to age-associated diseases. And one of the diseases I will focus on in the second part of my talk is bone diseases like osteoporosis, and we also have been working on skin aging in some context. So why do we believe that senescent cells are bad? Because mainly we think that an altered signaling function of senescent cells that persist in a human tissue, this altered signaling function that we call senescence-associated secretory phenotype, is involved in on the one hand driving tumorigenesis, but on the other hand also driving a pro-inflammatory status of tissues, where we have also a degradation of the extracellular matrix, and all problems of or many problems coming along with aging, and ultimately leading to depletion of regenerative potential, induction of pro-inflammatory phenotypes, and with that aging and age-associated diseases coming up. We also know that the latest biggest surprise in the field was that if we eliminate senescent cells from mouse models, at the end we can alleviate, improve, or even revert many of the most promising age-associated diseases, among them atherosclerosis, cardiac aging, diabetes, lung diseases, lung aging, and also bone diseases coming here along, bone and cartilage. So this was nicely summarized by João Basso's lab recently. And that's, as you all know, the mouse model that has been published by Beta de Kaiser, where the untreated mouse gets, maybe I can use a pointer that is better visible. Not sure if I can. Yeah, okay. Well, I think you can see my pointer anyway.
Yes, you can do right click and you can then find it.
Right, brilliant. Yeah, thank you. So, so we see the aged mouse, and then at the same age the mouse treated by the FOXO4 mimicking peptide that inhibits the FOXO4 p53 interaction, and by that removes senescent cells because they are dependent on this. So great promises here in the field. We have recently been interested in identifying proteins and genes and microRNAs that are differentially expressed in senescent versus non-senescent cells, and we tested then these genes and proteins if they would have effects on the lifespan of organisms as well. And two examples of these, one is a protein that is a splicing factor and at the same time a DNA repair factor called PRP19, or also SNEF. If we overexpress it in the fruit fly, we can actually indeed expand the lifespan a bit and improve the health span of these flies. The other example, if we knock down a protein, an enzyme that is involved in methylation of ribosomal RNA, we consider this as an epitranscriptomic modification or of the RNA of organisms. Also here we extend the lifespan of flies, of worms, and also of yeast after knockdown of this enzyme 5 protein, which, as I said, is a ribosomal RNA methyltransferase. So hitting only, modifying only a specific cytosine in the rRNA, having dramatic physiological effects then on the lifespan of individuals. So how did we run into extracellular vesicles? So the initial ideas go back to the work of Tom Rando's lab, where in a parabiosis model it was observed that the muscle stem cells in the aged mouse are kind of gaining function, of being rejuvenated, if you wish, by connecting the blood vessels to a young mouse. So this experiment suggested that there must be some factors in the blood of the young mouse that actively maintain stem cell functionality in the old mouse, or vice versa, that there are inhibitors in the systemic environment of the old mouse that are actively inhibiting stem cell functionality. And by now, a couple of different labs have shown that this rejuvenating effects can be seen in a couple of different organ systems when you have young systemic environment connected to the old mouse. And with this, we thought, well, something that would be very close to secreting something into the systemic environment might be senescent cells. We know they increase with age. and therefore the SASP might be a driver of these phenotypes. So we know that the SASP is built up by proteins, cytokines. We know that there are metalloproteinases, matrix metalloproteinases, a couple of different pro-inflammatory and matrix degrading activities, more than 90 proteins known to be bona fide SASP factors. There's upcoming evidence that lipids also form part of the SASP. And our question was, might extracellular vesicles and the microRNA cargo also be part of the SASP? So what are extracellular vesicles? By now there was a Babylonian language problem in the nomenclature of extracellular vesicles. Some people reference to them as microvesicles, then you had exosomes. By now the International Society of Extracellular Vesicles came up with the names according to the biogenesis of the vesicles. So if they are formed by the endoplasmic pathway by multivesicular bodies, which then fuse to the plasma membrane and release the vesicles into the interstitium, then we call them exosomes. If the vesicles bud from the plasma membrane, then they are called microvesicles. And if they come from apoptotic bodies, then they are, the old name of apoptotic bodies remains. So that's the three types of EVs that are forming the major classification. And if you refer to all of them, then you call them extracellular vesicles, which is the totality of the EVs that are secreted. By now we know that one donor cell secretes EVs, and then we have recipient cells taking them up. And the mechanisms of uptake, they are consisting of any uptake mechanism that has been published so far. It can be by endocytosis, by phagocytosis, or also by fusion of the EV membrane to the membrane of the target cell. Still, the biogenesis pathway doesn't give the EVs specific marker proteins. So there is still a problem in identifying ex vivo or in vitro specific subtypes of EVs. Still, therefore, size is used as a kind of a marker. So exosomes, the ones coming from the multivesicular bodies, they usually are between 30 and 120 nanometers in size. Microvesicles, those that are budding from the plasma membrane, they are usually a bit larger, 100, maybe 200 to nanometers to 1 micrometer in diameter. Apoptotic bodies are even larger, and then obviously we have the cells. So we consider these extracellular vesicles more or less like a bottle with a message in it. Evolutionarily, if you consider nothing in biology makes sense but in the light of evolution, evolutionarily it's hard to really come up with a good idea of where they originated from. I think most people in the field now think that EVs have been a garbage disposal mechanism in cells that then in multicellular organisms were somehow converted to a messaging system. And we have good evidence that definitely the EVs, they confer messages from one cell to another. And so that is clear. Might also be that EVs have been arising as a defense mechanism against viral infection. That is also one of the ideas of where they came from. So the question, what is on this letter? What is the message? And by now we know we have messages telling the recipient cells to grow, to proliferate, to perform angiogenesis, to modulate inflammation or the immune response. The EVs, they are known to be important for invasivity of cancer, epigenetic reprogramming, and preparing the metastatic niche. So it has been observed that a primary tumor sense EVs, they prepare the niche where the metastasis is forming, and by that, then the tumor cells can, after evasion to that site, can attach to that new site. What is the language of the letter? We have obviously all the biological proteins in there. We have lipids in the EV surface. We have proteins, and we have nucleic acids in form of sometimes DNA, RNA, mRNA, microRNA, even tRNA halves or ribosomal RNA, yRNA. So you find a whole bunch of different stuff in the extracellular vesicles, and it seems that all these different molecules within EVs are concertedly helping a recipient cell to change a specific behavior. So it seems to be a cocktail of a message, really a letter giving different commands to the recipient cells. My lab was mainly focused on microRNA in that context. And so the field of EVs is at the stage where we have Janus-phased extracellular vesicles, depending on where they come from. If the EVs come from disease target cells, or if they hit disease target cells, or if they come from diseased source cells, and I would add here senescent source cells, and among the different diseases I would place not as a disease but as a condition aging, then these extracellular vesicles might be like this famous other Austrian here, Christopher Waltz. They might be the bad guys in the system, while there's a very solid data by now that if extracellular vesicles come from mesenchymal stem cells or good source cells, and they hit good target cells, then they might more be like James Bond, actually almost like senolytics, helping to alleviate a lot of different, especially pro-inflammatory diseases, because it seems that the EVs have a specific immune modulatory functionality if they come from mesenchymal stem cells, for example. So one part that we were interested in was in skin aging, and so the idea was, do extracellular vesicles potentially coming from senescent fibroblasts, do they hit on keratinocytes? Do we see a crosstalk between these two cell types by extracellular vesicles? And so first we wanted to know if we find extracellular vesicles in human skin. So this is skin sections in transmission electron microscopy. We see multivesicular bodies with EVs in the skin. We see in immunogold labeling that we find markers of extracellular vesicles like CD81, like TSG101, CD63 in skin structures. We also find extracellular vesicle-like structures outside of cells, so that helps to think that these EVs are really secreted into the interstitium. And actually in cryo-EM we can also see them even better here, these nicely rounded EV-shaped forms surrounded outside with CD63, which is a membrane protein of extracellular vesicles. So this is 10, but can we also isolate extracellular vesicles from human skin? So we teamed up with Johannes research in Austria, who developed a method to to collect interstitial fluid from skin by an open flow microperfusion system. So we have a tube where we can where macromolecules and EVs can pass through. If we push buffer through the system, then we can collect in Eppendorf tubes the interstitial fluid of the skin. So we looked, so this actually is called a minimally invasive method. I'm not sure if this is minimally invasive to individuals volunteering for this. You can do the same method also in skin flaps, ex vivo. You can keep the skin flaps alive for about a week in the lab, and therefore you can also insert these microperfusion tubes. And then we characterized if there is extracellular vesicles in this interstitial fluid, and indeed it is, with the expected size and the expected shape in electron microscopy, and also positive in electron microscopy immunogold labeling when we used CD81 here. We also find some of the microRNAs that are our most favorite microRNAs due to some reason I don't expand on now. microRNA 23A, 21, 17, 31. So we find them in this interstitial fluid as well. So now if we consider now that there is a crosstalk, then the EVs from fibroblasts would need to go through an extracellular matrix, and for that we simulated this extracellular matrix with 3D organotypic skin cultures, where we use where we seed fibroblasts into a type 1 collagen matrix. These fibroblasts we pre-transfected with a C. elegans specific microRNA, CellMir39. This is not present in humans, so only in fibroblasts. Only in worms. So we put this into the collagen matrix. Then we seed keratinocytes on top of this. And then we let this skin equivalent differentiate at the air-liquid interface, which then gives you a very nice human skin-like structure with a stratum corneum on the top and the basal layer of keratinocytes. And here you see the fibroblasts in the collagen matrix. So now the question was, if we now separate the epidermal layer from the dermal layer, would we find this worm-specific microRNA also in the keratinocytes? And actually, this was really the case. So there must have been somehow a vesicular transfer of the microRNA from the fibroblasts to the keratinocytes here in the 3D model. So now, what are then the microRNAs, and what is the status of EV secretion in senescent cells? For that, we used stress-induced cellular senescence as a model, and then we collected the extracellular vesicles. They, in size and shape, they are the same as the vesicles collected from quiescent control fibroblasts. They express all the markers of extracellular vesicles, but now then this was the surprise. We find a 4 to 6 times higher secretion of extracellular vesicles per cell in senescent cells than in the quiescent control cells, which made us believe that extracellular vesicles indeed make part of the senescence-associated secretory phenotype, because that's what you find in the supernatant. Now, if we look at the microRNAs by a qPCR panel of about 800 different microRNAs, we find a lot of microRNAs upregulated in the senescent senescent fibroblast-derived vesicles. Among them, again, microRNA 23A here, which made us then perform some additional experiments. But first, we wanted to see if the senescent supernatants of fibroblasts would have an effect on keratinocytes in a 2D cell culture model. And for that, we performed the scratch assay, which is considered some kind of a wound healing assay. So we have a keratinocyte layer, we make a scratch, and then we add quiescent cell extracellular vesicles or senescent cell extracellular vesicles. And what we see is that the senescent cell-derived EVs, they promote the wound healing of the keratinocyte layer. So we can quantify that, it's significant. And what was surprising then, because this is a short-term effect, if we look at the long-term effect of senescent EVs on keratinocytes, then we observed that the differentiation, the terminal differentiation of the keratinocytes is not as good as with the quiescent cell EVs. So we have, in the first steps of wound healing, we seem to have an acceleration. Actually, this is in line with Marco de Maria's finding that you need senescence or acutely upcoming senescent cells for accelerating wound healing. But then for the last steps, it seems that chronic exposure to senescent cell-derived EVs doesn't really promote the nice terminal differentiation of the keratinocytes and the barrier formation at the end. Since we knew that miRNA 23A was one of the highly upregulated microRNAs in the senescent cell-derived EVs, we thought maybe miRNA 23A alone is doing this trick already. So we pre-transfected the keratinocytes with miRNA 23A, then performed the scratch assay, and actually we see a phenocopy of the effect of the EVs. miRNA 23A promotes the closure of the scratch, but it does not help to promote the final differentiation as here visualized with ecatherine and PKP4 as markers. So we think that miRNA 23A is one of the cargo molecules in senescent cell-derived EVs that drive this phenotype that we observed. In addition, when we looked at all the upregulated microRNAs, we had the impression that many of these microRNAs are involved in regulating apoptosis, actually in being anti-apoptotic. So we established an acute apoptosis model using acute oxidative stress on fibroblasts. So this is setting up the system. Staurosporin is inducing apoptosis in all the fibroblasts. This is without any stress. Extracellular vesicles don't really interfere with oxidative stress. So we induce apoptosis in the fibroblasts. And if we now use acute oxidative stress on the fibroblasts and quiescent cell-derived extracellular vesicles as control versus, oops, Microsoft PowerPoint was closed. Sorry. What happened?
Did your computer crashed?
No, the program crashed, but completely. Very funny.
Oh, no.
Sorry for the delay.
It's okay.
Should work again. Can you see it again?
No.
Okay. Now?
You need to share it again.
Ah. Mhm.
It's working.
All right. So sorry for the interruption.
It's in presenter mode. You just need to switch. Sorry. Perfect.
We indeed saw that the senescent cell-derived extracellular vesicles have an anti-apoptotic effect on the oxidative stress-induced apoptosis in fibroblasts. So we think that this is consistent with the idea that the SASP is something not wished for, because if you have damaged cells in the surrounding, then the senescent cells would protect them from undergoing apoptosis, while actually you would want these damaged cells to go into apoptosis due to potential genomic damage there. So there are papers claiming that, or there's actually one paper claiming that you have up to 50% of senescent cells in human geriatric skin. Most papers claim something up to 20%. Anyway, we decided that we wanted to model the presence of senescent cells in skin. So for that, we included stress-induced senescent cells, fibroblasts actually again, into our 3D model. So the fibroblasts were senescent, they are beta-gal positive. We can even re-isolate them from the collagen matrix afterwards to make sure that they were still there and they were still viable, so they reattach again, so morphology stays the same. The young fibroblasts look young, the senescent ones look senescent. Senescent cells don't proliferate anymore. So now if we add increased amounts of senescent cells into the collagen matrix, what we see is that we have a dose-dependent decrease in keratinocyte differentiation. And this is visualized by diminished epidermal thickening and epidermal thickness. As you can see, with 100% senescent cells in the skin equivalent, then you have a really very thin extracellular matrix. This comes along with a disturbed differentiation. So as we saw before in the 2D model where we had the senescent fibroblast-derived extracellular vesicles, we see something similar crosstalk of the fibroblast to the keratinocytes in the epidermis. So filagreen is as a marker of barrier formation and terminal differentiation. You see that this becomes spotty as opposed to the controls. We also see that almost half of the, more than half of the skin equivalents that we made got a defective barrier, so things can go in. The surface properties change when senescent fibroblasts are incorporated and you test only for the epidermal functioning. So, and all of these functions, they also are observed in the skin of elderly. So we have a barrier formation problem. So we have transdermal water loss in the elderly. We have a thinning of the epidermis in the elderly, and we know that the surface properties change. So we think that this model is a pretty close to human skin aging model. So the question now then, and we were working in a collaboration with Chanel in Paris. The question was, can we counteract using plant extracts? And actually we performed a screening of about 20 to 30 different plant extracts if they would revert some of the negative effects of senescent cells. And the Solidago Vigarea albestris extract was the winner. So the internal code of Chanel is 1201. So forget Chanel number, what is it, 7 or? So it's now 1201. So if we pre-treat senescent fibroblasts before putting them into the matrix in this 3D skin equivalent, we see that we indeed can rescue from this differentiation defect of the keratinocytes. So a pretty nice result that we got. Everybody was really happy, and Chanel seems to see similar effects in vivo in probants. We also tested for senolytic activity, and actually that was, yeah, well, we were not so happy because we obviously hoped that it would also be a strong senolytic. After 40 days on senescent cells, we start to see some minor senolytic effect. So this was, yeah, we thought maybe we can do better than that. And maybe we can identify a real druggable senolytic pathway. And for that, we performed some lipidomic analysis. And by this lipidomics, by mass spec, we identified two phospholipid species, lyso PPC and lyso SPC, so the formulas you can find here, as pretty strongly upregulated in senescent fibroblasts. So how does that look like? Lyso-PPCs in late passage in replicative senescent cells goes up. Lyso-SPC as well, P21 is high, beta-gal is up. In a stress-induced senescence model, we see lyso-PPCs go up. And if we use telomerase immortalized fibroblasts at the same population doubling level as we use the senescent cells as a serial passaging control for the cells in the replicative senescence model, we see that really lyso-PPC and SPC are upregulated only in the senescent cells and not in the long-term cultivated fibroblasts. So the lyso-PCs, they are a co-product of phosphatidylcholine cleavage by phospholipases. And if phospholipases cleave phosphatidylcholine, then the second product of the reaction is arachidonic acid. So the question was, first, are the enzymes that cleave phosphatidylcholines from the membrane, are they increased in senescent cells? And yes, all of them are increased, or many of them are increased. The activity of phospholipases is increased as well, and the arachidonic acid levels intracellularly in the cells are increased as well. So we thought, wow, nice. And then we knew from other fields that high levels of arachidonic acid are toxic to cells. So we thought, well, maybe if we block the conversion of arachidonic acid in senescent cells, this might be senolytic to some extent, which is a nice idea because you have a lot of small molecule drugs that inhibit arachidonic acid conversion, namely aspirin, diclofenac. They all block the COX enzymes that have arachidonic acid as a substrate and then make the prostaglandins. So using aspirin or diclofenac alone, or MK86, which is another inhibitor of the pathway, using them alone didn't really do the trick. But as soon as we combine drugs that stop arachidonic acid conversion in different arms of the pathways, COX1, COX2, or COX1 and reactivation of arachidonic acid, then suddenly we start to see that quiescent cells are not as much killed by this combination of drugs as senescent cells are. If we now take that to kidney epithelial cells that we made senescence, again using doxorubicin to induce stress-induced premature senescence, we can even increase the therapeutic windows to kill senescent cells by a factor of almost 400 difference in the drug, in the amount of drug that kills senescent cells, while leaving the quiescent kidney cells completely alone and did not kill it. So we think by this lipidomics approach, which is now, sorry, not microRNAs and not vesicles, but still I thought it was so interesting that I would add this data. So for the pathway, is this really due to the phospholipase activity? So the hypothesis now was that if we inhibit the phospholipase activity in senescent cells, then we would rescue senescent cells from this senolytic activity of the COX and FLAP inhibitors. And actually, this was really the case. So ASP 147080 is a inhibitor of phospholipase activity, and we can rescue the senolytic effect on senescent cells, while there are also activators and inducers of phospholipase activity available, and these made the quiescent cells more susceptible to the senolytic effect of our drugs. So with that, we think that we have a pathway. We have the phosphatidylcholine that are hydrolyzed by phospholipases that are upregulated in senescence. Then these are cleaved into lyso-PPC and SPC and arachidonic acid. Arachidonic acid is then converted by COX-1, 2, ALOX and CYP enzymes to eicosanoids, or they are reformed in the Lange cycle to arachidonyl coenzyme A, which then serves as a platform to rebuild the phosphatidylcholines. And if we block those by either triaxine C or aspirin, diclofenac, and similar, and combinations thereof, then the arachidonic acid levels increase to a point within the cell to induce apoptosis. And I think I used up my time and cannot tell you about the bone-specific microRNAs, which obviously I explained too long for each single slide, and with that, I jump to the acknowledgements and to the summary. So EVs and the microRNA cargo are part of the SASP and are potential biomarkers for age-associated diseases and senescence. And I think the most important answer that we are giving is that vampires, they bite young girls just to dilute the SASP. And which vampire would want to have a brittle skin for the rest of eternity? So I think that really is solved. The messages that senescent extracellular vesicles confer to other cells are to regulate apoptosis, differentiation, and proliferation. We see similar things that we see in the skin, we see in the bone, and that we can use plant extracts that act as xenomorphics to block SASP in senescent cells, and that the arachidonic acid conversion pathway is a novel senolytic target candidate. And with that, I would like to thank the people in my lab, specifically Ingo and Lucy, who were working on all the skin story. Then Tamerna alumni that went then into the company, like Lisa Schrammel, who was working on the bone story that I didn't tell you, and a fantastic new lab here at the LBI specifically Madhu Bobili, who is working on mesenchymal stem cell derived EVs to actually promote tissue regeneration in the elderly and in elderly preclinical models. And with that, many thanks for hosting me, and I'm very happy to take your questions.
Thank you very much. So if you want to ask a question, you can either write it in the chat or raise your hand. So I already have one question in the chat from David Gewirtz. He said, how did you distinguish between quiescent and senescent cells? I think it was at the beginning of your presentation.
Right. So the in our lab we were all, but the field was in the beginning when I was a PhD student, people in the field were using proliferating early passage cells as controls for senescent cells. And then obviously we realized that you always come up with cell cycle regulators that are differentially expressed between the senescent cells and the proliferating cells. So the field came up with the idea, instead of proliferating cells, to use quiescent cells, because actually that is what most cells in the body are in anyway. So it looks like it looked to many of us, it looked like a pretty good control. So how do we make cells quiescent? It really depends on the cell type. For fibroblasts, we usually wait until they are contact inhibited. Other people in the field, they do it by serum starvation. So they reduce the amount of serum that they add to the culture. So the cells also go into a quiescent state and cannot proliferate anymore. So we used contact inhibition. And how do we then differentiate? Well, first of all, it's they are in early passage. So they would still proliferate if we passage them. They do upregulate P21, but not to the levels of senescent cells. So we can differentiate still very nicely by P21 upregulation in the senescent cells. Also P16 is still a little bit higher in the senescent cells. And they are not getting as much as a beta-gal positive as the senescent cells do. And then obviously we can also look at HMGB1 and lamin expression and things like that as well. And senescent cells, we make sure that they don't proliferate after 3 weeks anymore. So when we have treated them with doxorubicin, we observe them and they just do not proliferate anymore, and we make sure of that also by BRDU assays.
Okay,
does that answer the question? Was a bit long, the answer, sorry.
It's okay. So now we have a question from Taranjit Singh. I'm sorry if I didn't pronounce well your name. I'm going to allow you to talk if you want to ask your question.
Thanks, Johannes. Really enjoyed your talk. Just a question. I was intrigued by the results of the extracellular vesicles on apoptosis. If I remember correctly, Manuel Serrano and Bill Keyes showed early in the embryonic development apoptosis and senescence cooperate. I'm sure Manuel is listening, so if I have got this wrong, please do tell. But maybe later down, this relationship of cooperation turns into non-cooperation. So I was just intrigued by your thoughts on that aspect.
Right. I cannot comment more really, or I cannot explain the difference. Obviously, it might be a very different thing to use the extracellular vesicles in the context of a stress-induced apoptosis model than in a natural developmental environment. And probably that is the major difference between the findings. And you could consider something like an antagonistic pleiotropy, that in the early development it's something positive, and and somehow by the setup of the tissue, then the EV suddenly start to behave anti-apoptotic. Manuel, I would be happy to if you're here, then what your take on it is.
Thanks, Johannes. I don't know if Manuel is here, but thank you.
We have a question from Chloe Reynard. Thank you for the very interesting talk. Did you look at the vesicles through TEM in other senescence models, in vitro or vivo?
Oh, sorry, I think I need to look at this question. I didn't really well. Could you repeat? Sorry.
Yes. Did you look at the vesicles through TEM in other senescence models, vitro
Yes.
or vivo?
Yes, we looked at TEM in vitro from the senescent cells. We looked in TEM and cryo-EM in human skin, but not yet in the elderly. So that is something that we are starting now. We didn't have the we didn't have sufficient samples at the time when we performed the study, and our PhD student had to rise up and finish. So, but this is planned that we also do it now in the skin of elderly. But it's not to be expected that the shape or size of extracellular vesicles would be different, but we would definitely want to quantitate if we also find more EVs in the interstitium there. And the main experiment actually that we are going for is this isolation of the interstitial fluid by the open microperfusion system, because that is quantifiable better, and we can also look at the what the cargo of the EVs ex vivo would be.
So we have Manuel Serrano who can.
Hi, Johannes. Congratulations for the fantastic talk. I really enjoyed it. It was very nice.
Thanks.
Actually, I had a question. Well, it's more a commentary. I don't follow closely all the progress on the extracellular vesicles and senescence, and I wanted to ask you what is known about these extracellular vesicles in vivo. For example, in diseases like fibrotic diseases, the ones you mentioned in the beginning. Is there any evidence that, well, not evidence, can these EVs be detected, EVs with senescent features?
The answer is we don't really know yet. So the EV field is mainly focusing on using them as a therapeutic, obviously from MSCs. And in those cases, the MSC-derived EVs definitely help in preclinical models against fibrotic diseases. So there are a couple of papers out there that show that. For senescent cell EVs, if they really do some harm, it's not really been done. And what we have started with in collaboration with Dave Munro in Mayo is that we place a recombinant membrane protein of EVs, so CD81 plus a affinity purifiable entity with a tag. We place that under the P16 promoter that they are also using in the INCA mouse model. And with that, we think that we will be able to identify senescent cell-derived EVs in the tissues and see if they would then really accumulate at sites where we have age-associated diseases and an increase in senescent cells. But so far, otherwise, the tools are not really there to differentiate senescent cell-derived EVs in vivo yet. It's not well done.
Mm-hmm. Thank you. Thank you.
And one last question from Violeta. How were the devices that collect EVs from the skin implanted?
How was the device?
Yes.
That
collected the EVs.
It's called open microperfusion system. It has been developed for skin penetration studies, actually. By friends in Austria, in Graz. I'm just going to that slide and show it again. So it's a custom custom-made techniques. Joanneum is also is not only a scientific institution, but they are also acting as a contract research organization for in the field of dermatology, and they are using that for showing that there is penetration of a substance or a drug to the different layers of the skin. And what they can do by ultrasound, they have an extremely sensitive ultrasound system, so they can make sure that the open microperfusion tube is placed at a specific height in the skin, so that it's really either in the epidermis or at the border to the dermis, or within the dermis, or even in the hypodermis, where you have the adipocytes and the pre-adipocytes. So, did that answer the question?
I guess. We have a question from Effie Tsuko. How about timing and immune system clearance? Effie, maybe if you want to explain, I can allow you to talk. Maybe the context.
Hi, guys. Yeah, great talk. I was wondering actually whether, because you said that it's difficult to have the tools to see the clearance sometimes or detect the cells. Have you had any macrophage or immune cells being more present or not when you use this analytics, etc.? And when would you check for this, if you have done any of the work?
Well, actually, we have no real good data yet on that, not in vivo. In vitro, what we did in terms of recruiting macrophages or in altering macrophage behavior was that we used the lipids, because they would be the surface of the EVs. And if we have more lyso-PPC and lyso-PC on senescent cell-derived EVs, or if we have in total more of these molecules, because the senescent cells secrete just more of them, then the question was really, what do the macrophages do with that? So we used the macrophage cell line in vitro, which is not a good model, by the way, but still we tried that. And what we see there is that these lyso-PCs on their own, they seem to inhibit the phagocytosis of the macrophages. So it seems that these molecules, these lipids, these phospholipids, that would increase. So no proof of that yet. It's a speculation that this could happen in vivo as well. But it could be that these lipids secreted within extracellular vesicles, that they inhibit the phagocytosis of the macrophages, and by that are one contributing factor to senescent cells being able to chronically accumulate in tissues.
So it's kind of like blunting the response, in other words. Similar to cancer cells, then. We should think about them more like that.
An experiment that definitely should be done. We will not do it with cancer cells. But yes, it would be, we would expect that this a similar, or there could be a similar mechanism in cancer cells. We know there are a couple of mechanisms how cancer cells dampen the immune system. This would be an additional one, and yeah, could happen. But in senescent cells, it could also be some one of these So with all these pro-inflammatory factors, look, immune system, we are here, but then me as a senescent cells, don't touch me. Oh no. So look, don't touch concept maybe.
Mm-hmm. And maybe a last question from Yatsu. Are miRNAs stable in EVs?
Yes, they are stable in EVs. We can use them. That's the part I didn't show you. We can use blood samples from 20 years ago if they have been stored at -80. They can even be frozen and thawed a couple of times. So the EVs really protect microRNAs from degradation. If you spike microRNAs in human serum, it's gone because you have enough RNase activity there. But within the protective casing of the bottle of the message, they are stable. And actually, I didn't have time to explain in detail the experiment, but we for the microRNA 23 that is upregulated in the supernatant or in the extracellular vesicles of senescent cells. We also used RNase treatment, and we saw that around half of the microRNA is still protected. And if we then add detergent that opens up the membranes of the EVs, then we digest away all the microRNA. So there is definitely part of at least some of the microRNAs that are inside of the extracellular vesicles, but there is also part of microRNAs that are probably, or well, it's known that they are also associated to proteins in the supernatant.
Okay, thank you very much for this very interesting talk. And so if someone wants to present a talk next year, don't forget to contact us. We have also openings in yICSA association. And I wish everyone a nice end of year season, and see you next year.
Thank you. Thanks again.